Just a second. Okay. So can everyone see my screen? Is it visible?
Yes.
Okay. Thanks, Kshitij. So guys, I would request all of you to please mute your microphones if that's okay, so that we have less disturbance. Just raise your hand or unmute when I ask a question, okay? I'll ask a lot of questions, presuming that you have read the chapter. So I'll begin. So it's the chapter 28. Most of my resources are from Harper's, but I've really tried to add in all the missing points or points that are really high yield or important. So we'll begin. So first, the biomedical importance. It is just talking about the nitrogen balance in our body, okay? So nitrogen balance can be either nil or zero, or there can be a positive nitrogen balance or a negative one. So anyone would want to tell me what is a negative nitrogen balance? Anyone? Just volunteer or I'll call you out. Anyone? Kshitij, would you want to enlighten us? Just a short single line answer.
I think it's loss of.
Okay, Anisha, go ahead.
Negative nitrogen balance, that means the intake of nitrogen is more than the output.
Is it? Does everyone agree with that? Is that negative or positive?
Sorry, that's positive, and negative one is the output is more than the intake.
Exactly. So in simple terms, when you are taking more nitrogen and excreting less, that's a positive one. And if you're excreting more nitrogen than what is being ingested, it's a negative one. So in simple terms, negative nitrogen balance would occur in states of when your body is not able to cope up, okay? Like states of when you have a surgery or you have malnutrition or cancer, something like that, okay, which is not good. Whereas a positive would mean something nice, right? So when you go to the gym and you build your body, so that would help you gain muscle. That's a positive nitrogen balance. When you're pregnant, the uterus muscles grow in size to support you, right? So that's again a positive one. These are all examples from your book. Okay, moving on to the next one. So protein turnover, it's nothing but the amount degraded and regenerated, okay? So just remember that it's 1 to 2% each day of the whole total body protein. Again, I'm just highlighting the main points. I won't be telling you everything in the book. Next, moving on, how protein degradation takes part. So in this one, I bet everyone knows what half time is. Anything from full when it goes till half, the time that it takes is nothing but the half time, okay? So just remember that the housekeeping enzymes and the key regulatory enzymes, this one would be the half time for them, okay? Main takeaway point from the slide, it's the pest mnemonic. It's for proline, glutamate, serine, and threonine. So these are the target proteins for rapid degradation, okay? So they target, what are proteins? They are nothing but made up of amino acids, right? They make up the proteins. So just remember this one. So in this, we'll talk about two. ATP independent degradation, obviously, that would not require a lot of energy, right? No energy, in fact. And the most important one, I want you to really take note of this one, because it will definitely come out in the exam tomorrow. So ATP and ubiquitin dependent degradation. So this one, I want you to focus in which part, what reactions take place, okay? So what are we doing in the cytosol? So what happens in the cytosol is the degradation of the proteins that have a short half-life or those are abnormal. It occurs over there. Okay, obviously something that is misfolded or not normal, it will degrade quickly, right? And hence it will have a short life because it's abnormally encoded. So it would require energy. Our body is trying to fight something that should not be there. So we are spending extra energy on that, right? So hence it will require ATP and a very special molecule called the ubiquitin. So some things about the ubiquitin, guys. Derek, can you please mute yourself? Okay, thank you. So in ubiquitin, it targets the protein for degradation. So things that I want you to know from this, it's that the amino terminal would depend whether the process would retard or accelerate. Okay. So methionine and serine are the ones that retard, and aspartate and arginine are the ones that accelerate. You can just simply remember by the A's. A is for accelerate. So automatically by elimination, you can remember that other two are the ones that retard. Okay. So what happens in polyubiquitination? So it's like I'll show you this diagram. Okay. Coming here, what happens? There is involvement of the lysine residues. Just remember these numbers, lysine 63 and lysine 48. That's what I have. So there's the attachment of 4 or additional ubiquitins and these lysyl residues, okay? So next, important, very important, I've marked a Q. Take note on the white paper you have. So in this, what happens basically is you could be asked, like, where does the degradation of ubiquitin tagged proteins take place? So examples could be cytosol or a proteasome or a mitochondria. So what would be the answer? Proteasome, okay? This one has the answer in it. The next slides don't. So better have Google or book by your side because you are the one telling me the answers. So moving forward, this is a three-dimensional structure of ubiquitin. And here are some key disorders that are related with anything that goes wrong with ubiquitin or the enzymes related to ubiquitin. So you should probably note these down. They can again come as a like following cause ubiquitin disorders except like so Angelman syndrome, autosomal recessive juvenile Parkinson's disease, von Hippel-Lindau syndrome. This one is really interesting. You can read about this. And congenital polycythemia. Okay, and here is the proteasome where the degradation of the tag protein was taking place. So in this, it consists of a macromolecular cylindrical complex of proteins. Okay, so they are stacked like donuts. So obviously they'll be like an inner ring and an outer ring. Okay, so in the central for degradation, where does the protein enter? You can all see the ubiquitin is entering in the central core. Okay, so again, it is regulated by the two outer rings. Those are whose main function is to recognize the polyubiquitinated proteins. So this is nothing but a very simplified diagram of how it works. So as it enters the central core, it keeps on degrading, and this is the end product. Okay, everyone clear with that? Any doubts till now? Am I going too fast? And okay, fine. So next. Oh, there's a question. So in inter-organ exchange, it's the one that maintains the circulating levels of amino acids. So we all know that there are different amino acids released by different organs, right? As mentioned in the book. So mainly we are going to talk about muscle because it's the one that generates more than half of the total pool of free amino acids. Okay, so there are three that I really want you to remember. You can remember it as GAVE. The muscle gave 3 amino acids. Okay, like GAVE. So glutamine, alanine, and valine. So they come from the gut and kidney. You can see muscle. This is gut and kidney. Okay, this one. And alanine goes this one to this one is the alanine over here. And the only two main organs that play a role in the circulating amino acid levels are the muscle and the liver. So And you would notice one thing in the diagram that only valine is the one going to the brain. It's a branch chain amino acid, right? So it is the one taken by the brain. What are the other two branch chain amino acids apart from valine? Come on, quick. Google it if you want. I want the answers from you guys. There are 3 branch chain amino acids. One is valine. What are the other two? Come on, guys. You can use your Google if you want. Just have to search an answer. Anyone?
Leucine, isoleucine.
Yes, that's great. Who is the one who answered? Can you raise your hand? Okay, 007. I don't know your name, but yeah, that's correct. So leucine and isoleucine are the other two branch chain amino acid. You can remember that by a mnemonic called LIV, L-I-V. And the next question, which amongst the amino acids do you think is the key glucogenic amino acid? Very important. Again, take note. It might come in your exam. It's very important. It's even mentioned in this slide if you want a hint.
Alanine.
Exactly. So alanine is the key glucogenic amino acid. Take note of that, really. So moving on to the next part, this is how the flow works, okay? Remember that the key, take down note of 2 points. I'll be telling that. It's not mentioned in the presentation. From all our cells, glutamine is the one that is released finally, majorly. And from the muscles, it is alanine. Okay, so there are only two ways the amino acid comes to the liver, and that's by alanine from the muscle, glutamine from all the cells in our body. Okay, so in this, what happens, you can see that in the muscle, the glucose is there, then the transamination reaction takes place, and alanine is formed, and alanine goes to the liver, and again the whole cycle goes on. Okay, so in this, we should always take note of the fed state, and because the physiology matters. So we are talking immediately after eating. So what happens in the gut? The 20% of the branch chain amino acids, they go via the portal circulation to the liver. And again, from the liver, the 60% would go to the muscle for the whole cycle to continue, because alanine is coming from the muscle, you can see. Okay, so what happens after eating a protein-rich meal? Take note of 2 points. The splanchnic tissue, this one, it releases the amino acid, and the peripheral muscles, they extract the amino acid and send it back. Okay. Next. So in this, we would take note that animals convert alpha amino nitrogen to varied end products. So when there is excess nitrogen, we can classify it as ammonotelic organisms, ureotelic organisms, or uricotelic. I bet we all read this in our 12th class or 11th class. So ammonia, can anyone tell me which organisms release ammonia? Just one line, quick.
Fishes.
Exactly, fishes. So just remember the examples. We just want to focus on urea. How about uric acid?
Birds.
Birds.
Exactly.
Birds.
And urea, it's us mammals.
Humans.
Okay? Humans, exactly. So we have to take note that urea is a non-toxic form, whereas ammonia is a highly toxic form. Okay, we cannot find ammonia. Just think that ammonia is like a prisoner or a thief. Okay, and in our body, we always require a policeman to be with that thief. So ammonia would never be alone until it reaches the liver, which is like the prison. So the policeman is all the amino acids that are helping to carry ammonia to the liver. Everyone clear with that? It's the simplified way of how I can put it for you. Because ammonia is highly, highly toxic. It cannot exist freely in our body until it is in the liver, because liver is the only place where the urea cycle can take place. Okay. Everyone clear with that? That's the simplest form I can put it with. Give me a thumbs up. It's okay. Don't unmute yourselves. Okay, cool. So next, we'll talk how urea is formed. This is like the whole slide. If you understand this, you will understand the rest of the whole session. So what happens in the biosynthesis of urea? Remember, urea, U-R-E-A. So 4 letters, 4 stages. So what happens in the first stage? It's transamination. So transamination is nothing but when the amino group from one amino acid goes to the other amino acid. So you can see here, there's an alpha amino acid. So when you release the ammonia or the nitrogen group, you are left with the keto bond. Hence, it is called an alpha keto acid. Now, the nitrogen that was released here would join the alpha ketoglutarate. So here the nitrogen joined, it gave us another amino acid. Make sense? It's like give and take at one point, okay? Makes sense, right? Nitrogen goes from here and it forms the alpha keto acid. Nitrogen joins the alpha ketoglutarate and gives us glutamate. Okay. So next would be the oxidative deamination of glutamate. Remember, guys, glutamate is the only amino. Again, write this down. It is the only amino acid that can undergo oxidative deamination. None of the amino acids can. Okay. Hence, everything that reaches liver is ultimately converted to glutamate, because that is the one that will release the ammonium ion and send the ammonia into the urea cycle. Okay. So take note of this point. You can see, right? Glutamate undergoes oxidative deamination. It releases the ammonia, converts back into alpha ketoglutarate because it gave the ammonia. And now the ammonia enters the urea cycle, combines with CO2 to form urea. Okay, so next would be the ammonia transport. I explained you that ammonia cannot exist alone. It is highly, highly, highly toxic. I cannot stress enough on that. Okay, so there has to be a policeman with the ammonia to help it reach the prison. That is the liver. Okay, so last would be the reactions of the urea cycle. There are 5 enzymes in the urea cycle. Each is very important. Each one has a disorder associated with it. Okay, so moving forward, this slide is all questions. Okay, so transamination, I already told you what happens in transamination. Alpha amino acid gives out its nitrogen. Nitrogen goes out, it forms a alpha keto acid. Then the nitrogen combines with the alpha ketoglutarate and it forms this one, glutamate. Okay, so I want someone to tell all the amino acids except there are 4 amino acids mentioned in your Harper's. It's a very potential MCQ, guys. Anything that is in a that is 4 can be an MCQ. Anyone who can give me the 4 amino acids mentioned in your book that do not undergo transamination?
Proline and
Life.
lysine are two.
Proline, hydroxyproline.
Threonine, hydroxyproline.
Okay, so it's proline, threonine, hydroxyproline, and the 4th one is
Lysine.
lysine. Yes, exactly. So take note of that. I have not included that. Next, addition of the substrate and release of product. What is this mechanism called? Again, a very potential question for your exam. I'll give you a hint. It's one of the sports we play. Anyone? What
Ping
is
pong mechanism.
exactly? Great job, Mansi. I'm so sorry.
Yes, correct.
Okay. Not included in your book, but can anyone give me other two names for the ping pong mechanism? Anyone? What is the ping pong mechanism also known as? No one? Okay, I'll give this to you. So ping pong mechanism is also called a bi-bi reaction because two things are happening. It is also called a two product, two substrate reaction. Okay, you can note this down. It was not there in your book, so I added that. Next, it is present at the catalytic site of all aminotransferases, and it plays a key role in catalysis. What is this molecule called as? I'll give you a hint. Vitamin B6 is very, very essential for this molecule.
Pyridoxal phosphate.
Exactly. Great job, Wilson. So pyridoxal phosphate is very important for all transamination. Without that, there cannot be any reaction. So vitamin B6, it's also called as, what's the name for vitamin B6?
Pyridoxine.
Exactly. So hence, it's very important for enzymes to function properly in our body, okay? So pyridoxal phosphate, please take note of that. Next, moving forward, it was the oxidative deamination of glutamate. Guys, this is very, very important. Please take down note of the enzyme involved in this reaction. So it is L-glutamate dehydrogenase. Okay, so it is the one that is really very important for the nitrogen metabolism. Take note of these. It is inhibited by ATP, GDP, and NADH, and it is activated by ADP. So I want you to take note that all the inhibition one are these are the like ADP for, these are like the products, but this one is the substrate. Just note down that these are products and it's activated by a substrate. Okay. So really take note of this one. You can see the thing I was telling you in the first place, that glutamate is formed by the ammonia. This is transamination reaction again. So this one take note. Next, amino acid oxidases remove nitrogen as ammonia. So in this, nothing but the amino acid is again changing into an alpha keto acid with the removal of the ammonium ion. And in the end, catalase is the one that's removing. Catalase is nothing but it from hydrogen peroxide, it gives out water and again oxygen. This one, not so important. Moving forward, this one I really want you to focus because it's conceptually very important. Ammonia intoxication. So why do I keep saying that ammonia is highly, highly toxic to our body? So first of all, where does ammonia come from? It comes from the enteric bacteria that are there in our gut and the tissues. And then again, I told it's transported to the liver, and from the liver, it goes and forms urea. Okay. So I have given the answer to my own question here, but still I'll try to explain it better. So ammonia, it is majorly toxic to the central nervous system, and anything that affects the brain would affect our whole body. Everyone agrees with me. The most severe thing that can happen is something affecting the brain, okay? So what basically happens, it reacts with alpha ketoglutarate to again form the glutamate. Always it tries to go into the glutamate form. Okay, so whenever the alpha ketoglutarate is going to form glutamate, the alpha ketoglutarate levels will start falling down. So what will happen when they start falling down? Your Krebs cycle or the TCA cycle in the neurons that is happening, they will get depleted of what? what is your TCA cycle for? Anyone? What's the end result of your TCA cycle? Guys, what does the TCA cycle give you?
ATPs, GTP.
Exactly. ATP is nothing but the energy. So if you don't have the basic molecule to give you ATPs, your brain, levels of ATP in the brain will start going down. Okay. So this I'll explain it in the next slide. And the portal blood bypasses the liver. So also it causes ammonia intoxication. Examples, it happen in cirrhosis, or when extra blood vessels form collaterals, then also it will bypass the liver, and we do not want it to bypass the liver, because then it will go directly in the bloodstream and it can cause tremors or slurred speech or blurred vision. Okay, so we don't want any of that. So okay, this is the main thing, simplified. So what basically happens, why is ammonia toxic to brain? Okay, so generally when the ammonia, normally in us, ammonia would combine with the hydrogen ion and it will form the NH4 plus. This NH4 plus, it cannot cross the blood-brain barrier, okay? But NH3 can. So when NH3 starts rising, not enough hydrogen ions can get together with NH3 to form NH4. So what will happen? This will cross the blood-brain barrier. And I told you, anything that adds nitrogen to a alpha keto group, it will form an amino acid. Do you get me? So what happens? Excess ammonia is there. So it combined with alpha ketoglutamate to form the glutamate. Okay. So then glutamate went this enzyme, glutamine synthetase. Okay, guys, interesting question. Can anyone tell me the difference between synthetase and synthase? Anyone? What's the difference between a synthetase and synthase?
I guess that lies in whether the enzyme is using ATP or no.
Exactly. So which one is using ATP?
Synthetase.
Yes. So this one is an energy requiring process. So when this happens, there is more ammonia. So there is more of alpha ketoglutarate changing into glutamate. Okay. So this glutamate then changes into glutamine via the glutamine synthetase. Okay. So as the levels increase here, more of this is formed, glutamine levels will start rising. So why is it getting toxic to the brain? So firstly and foremost, the glutamine increase would cause the water to come in. We all know that ammonia molecules, this is majorly the process of osmosis going on here. So nitrogen this has 2 nitrogen molecules, whereas this only has 1. So this will attract water to it, and this will cause the brain to swell up. So this will cause the cerebral edema. So next would be the glutamate. So what is happening over here? It will undergo decarboxylation. Why? Because it's giving out a CO2 molecule. So when it gives out a CO2 molecule, it will cause increase in alpha gamma acetobutyric acid. Okay, we all know that it's an inhibitory neurotransmitter. So this one, since it's increasing, it will cause CNS depression. Okay, bad, very bad for the brain. This should not be happening at all. Okay, but now I told all of this was going into glutamate. So this is the one I explained in the previous slide. There is decrease in alpha ketoglutarate and there is decrease in the TCA cycle, and hence finally there is decrease in the ATP production. So there are 3 things that are happening. So depletion of ATP, CNS depression, and cerebral edema. Okay. Take note of these 3 things that are causing the toxicity to the brain. Next, moving forward, I just now told you about the glutamine synthetase enzyme. So always remember, when there's ammonia in the body, this one is the first one to come and trap it so that it does not harm you. Okay. So question, where is glutamine synthetase found in your body? Which organelle?
Mitochondria.
Yeah, mitochondria. Take note of this one. It's very, very, very important. So what does it do? It fixes ammonia. Look here. The ammonia came to your cell. The glutamine synthetase, it took the glutamate, formed the bond to give glutamine. Told you this one only has a single nitrogen. This one has 2 nitrogens. This is the form that can go into your blood. So glutamine goes to your blood and to your liver. Then glutamine is again converted back to glutamate. Okay. So it has a major role in ammonia detoxification and acid-base homeostasis. Remember this line from this whole, it is the first line trapping of ammonia. Okay. Moving forward, we'll talk about the glutaminase and asparaginase to deaminate glutamine and asparagine. So again, I told you, right? Glutamine is the one that's helping to carry ammonia via blood, because it is the one that can travel in blood. And it will take it to the liver, where it will again get converted to glutamate. We are still having, do you get the whole process? Glutamine carried the ammonia to the liver so that it can safely remove the ammonia, because liver is the only place where ammonia can exist freely. Is everyone clear with that? I cannot stress enough on that. So we have 2 types of glutaminase. This one is the major one. So hepatic levels rise in response to high protein intake, because when you are taking more amounts of protein, there will be more amino acid formed, more amount of ammonia formed. Hence, the enzymes also should increase in quantity. And in renal type glutaminase, there is increase in metabolic acidosis. So can you all focus on this, my pointer for now? So this is what happened. Glutamine, the glutaminase, it gave out glutamate again and released ammonia, which will go into the urea cycle. This is the same thing that's given in your book. Next. Spare my bad handwriting. So in this one, so urea is the major end product of nitrogen catabolism in humans. We all agree, because that is the only way we can excrete nitrogen. So in urea biosynthesis, I hope you all went through the urea cycle. That's the main takeaway from today's lecture. So first thing, there are 6 amino acids taking part. In the first step, this molecule, it functions solely as an enzyme activator. So what is that molecule? Without it, the urea cycle will not move forward. I'm again stressing, it's a molecule, it's not an enzyme.
Ammonium ion.
N-acetyl glutamate.
Yes. Who is the one who answered? Can you raise your hand? Okay, Anisha, yes, it's the N-acetyl glutamate. It's an obligatory molecule. Without it, the carbamyl phosphate synthetase 1 will not function at all. Okay, so it has to be there. Any deficiency in this molecule will cause a buildup of ammonia. Okay, so now our end product is to form. Can someone tell me what is this molecule I have drawn over here? Do you recognize this molecule? Anyone? Can you guess? Just don't focus on these arrows. Just this center part. What is this molecule called?
Urea.
Yes, urea. It's written here. So I just want you to remember that in the urea cycle, it is just the ammonium ion. Look over here at my pointer. The ammonium ion, the CO2, and obviously energy. Aspartate. Aspartate is the one giving this second nitrogen. So these are the ones that are consumed. Rest, everything is regenerated. So this one was in your book. So we use 3 moles of ATP, 1 mole each of ammonia and aspartate. Okay. Next, coming to the big part. Okay, what is the cycle called as?
Urea cycle or ornithine cycle?
Okay, urea cycle or ornithine cycle. There's one more name to this cycle after the scientist who discovered it. It came out for me in my exam. Maybe it might come out for yours. Can someone tell me the other?
Krebs-Hanslet cycle.
Yes, Anuj. It's the Krebs-Hanslet cycle, named after the scientist who discovered it. Very, very, very important. So we'll go step by step, okay? So in this one, obviously the first step would always be the rate limiting step. So what would be the rate limiting enzyme? It is, it's okay, unmute and say, it's all on the screen.
phosphate.
Synthetase 1, yes. Again, synthetase because it is consuming ATP. I want you to take down note of the ATP, the number of ATPs. So this enzyme uses 2 ATP. It will help you after, okay? So, okay. So the next thing I want to ask, what are the reactions that are occurring in the liver mitochondria? And what are the reactions occurring in the liver cytosol? It's all on the screen. I'm just seeing if you're actually.
Reaction
1 and 2
One and
happen in the mitochondria.
Yes, and all the rest happening in the liver cytosol. So
Liver cytosol.
remember that urea cycle is one of the cycles that takes place at 2 places. There are more. Try to look for them. It will be a question in the last slide. So first thing, we took note that CO2 ammonia that came from the aspartate or the glutamate, this one is providing the first nitrogen for urea. Okay, so in this one, the carbamoyl phosphate synthetase 1 or the CPS1, it combined with the ammonia and it formed the carbamoyl phosphate. So now what happens is the carbamoyl phosphate would combine with ornithine. Guys, I want you to take note, ornithine is not present in the mitochondria. You see the little red dots over here? Can someone tell me what these red dots are? What are these red dots on the mitochondria?
Carrier proteins? Or
Yes, they're the carrier proteins. I think you read your book really well. Okay, so these are the carrier proteins. So what happens is the ornithine is actually not inside the mitochondria. So when this one is outside and this little red dot helps it to bring inside, because this reaction can only take place in the mitochondria. So it combines with the second enzyme, the ornithine carbamoyl transferase. So again, no ATP, take note, in the second step, no ATP is used. So this one just transfers the amino group to the ornithine and it forms the citrulline. Okay. Next, coming to the third step. So in the third step, now the citrulline and the aspartate. I told you in the first itself that we need 2 nitrogens for the urea to form. 1 nitrogen is from the ammonia itself. The second nitrogen is from aspartate. See this blue highlighted portions. So when citrulline combined with aspartate, using the enzyme arginosuccinate synthetase, focusing again, it gives us arginosuccinate. You can see this single blue thing now has turned into 2 nitrogen atoms being highlighted. So if a question is asked to you, what is the source of the nitrogen in the final product of urea cycle? 2, ammonia and aspartate. Okay. So again, we used 2 ATPs over here. Now this again, we used 1 ATP over here. I hope everyone is taking note, because you can be asked about the energy consumed in the urea cycle. So in the third step, we used an ATP. Moving forward, in arginosuccinate, now we all know that lyase is used to split. Okay, so arginosuccinate, it gives out arginine and fumarate. Can tell me fumarate is linking urea cycle to which other cycle?
Krebs cycle.
Exactly. So fumarate goes to the Krebs cycle to again, I want you to focus over here really. So fumarate, it with the water molecule, it forms malate, and then this malate, this enzyme malate dehydrogenase, it gives us oxaloacetate. So with the help of the enzyme AST, oxaloacetate is again converted to aspartate. Do you see the link between this one Krebs cycle and urea cycle? It is again regenerated so that it can continue the urea cycle, and the glutamate is converted into alpha ketoglutarate. So just take note of the links between the cycles. It's very, very important. So fate of fumarate, we all got to know, it will regenerate aspartate by transamination. Okay, so now we have the arginine. Now the arginine would finally give out urea via the enzyme called arginase in the 5th step. Okay, so arginine and again ornithine would be regenerated. Like one of you mentioned that it's the ornithine cycle. Why is it called the ornithine cycle? It is because the ornithine gets regenerated. Okay, everything is clear. Everyone is clear with this one, because we'll be talking about the enzyme deficiencies and what disorders they will. Give me a thumbs up so that I know I can proceed forward. Okay. So one thing to take note over here, ornithine and lysine, they are potent inhibitors of arginase. Obviously, if there is ornithine buildup or no ornithine forming over here, the arginase would not be functioning. Can anyone tell me where does this lysine come from? I didn't mention lysine until now in the whole cycle. What is the role of lysine here? It's mentioned, it's a line from, I copy pasted this from the book. Did anyone try to figure out why was lysine there? Okay, take it as an assignment, do it later. Find out why the lysine inhibits arginase. Okay. And also take note of this sentence that it serves as a precursor of a potent relaxant that's called nitric oxide in a calcium dependent reaction, and it's catalyzed by obviously nitric oxide synthetase. Again, it's a energy requiring process. So moving on, I'll try to summarize it now. So I told you now, all amino acids are metabolized into a nitrogen that would give out ammonia, okay, except for aspartate. Always take note of the exceptions. And in this one, I told you aspartate, ammonia, they would enter they would give the urea, enter to the urea cycle, and the urea is formed by the urea cycle. I'm really sorry. And the kidney would excrete out urea. There's a statement written in the book that, urea buildup is there when the kidney is not functioning properly, and it's not vice versa. Buildup of urea never causes kidney dysfunction, but kidney dysfunction causes urea buildup. Could everyone understand that? Because urea is a very, very, very non-toxic molecule. So even if it is high in levels, it would not cause kidney damage. So urea buildup is a result of a kidney pathology, but kidney pathology is not due to urea buildup. Okay, just take note of this. It was there in your book. Okay, and remember about N-acetyl glutamate. It is very important for CPS1 to function because it's the one that helps ammonia to bind and form carbamyl phosphate. Okay, so next, very important topic, the metabolic disorders of the urea cycle. So, okay, let's begin. So you have to remember there are three main features of anything going wrong with the urea cycle. Hyperammonemia, obviously there would be buildup of ammonia, so there will be hyperammonemia. There will be encephalopathy. I explained you why. Okay, it is because of the toxicity due to the CNS. And respiratory alkalosis. Can someone enlighten me why there is respiratory alkalosis in the patients? First of all, what is respiratory alkalosis? Is it a derangement in bicarb or CO2?
CO2.
CO2.
CO2, right? Respiratory. And what happens to the levels of CO2 in respiratory alkalosis?
Decreases.
Decrease.
Exactly. So why is this happening? So remember, guys, whenever there is buildup of ammonia in your body, the patient would be tachypnic. That is one of the differentials you should always take down note of a urea cycle disorder if the patient coming to you is tachypnic. Always try to rule out a urea cycle disorder. So when there's tachypnea, you're breathing really fast, right? So you're letting out a lot of carbon dioxide. So what happens when you let down a lot of carbon dioxide? The levels of CO2 would decrease, right? Levels of CO2 decreasing is the one causing the alkalosis. Are you able to relate now? So we can agree that we know why hyperammonemia is there, why encephalopathy is there, and why respiratory alkalosis is there. Along with this, you would also see that the child would be vomiting. They would avoid high protein foods. There will be intermittent ataxia, irritability, lethargy, and in severe cases, there will be mental retardation. Okay, so now what should we do? How do we manage? So in the treatment, we would give the patient a low protein diet because we still need those amino acids. So we still need protein. We cannot stop having proteins. So we would go low protein diet so that the enzymes are not overwhelmed processing those proteins. Okay. Hence, we would not avoid this sudden increase in blood ammonia levels. So remember, it can, in the exam, they can ask you for the treatment of ammonia disorders, a high protein diet is recommended. True or false?
False.
False.
Exactly. So, but our goal still remains that we should provide sufficient protein, especially arginine, and energy to promote growth and development while minimizing the metabolic repercussions that can be there. Okay. So, guys, the next slide, try to draw it on a piece of paper. We'll be listing down the disorders that are associated with these 5 enzymes, okay? CPS1, ornithine carbamyl transferase, the arginosuccinate synthetase, arginosuccinate lyase, and last arginase, okay? So please go step by step and draw this with me, okay? So first, we start with CO2 and ammonia, right? So CPS1, along with the N-acetyl glutamate, it gives us carbamyl phosphate. So if this enzyme is deficient, there will be primary buildup of ammonia, right? Direct toxicity is there. So there will be hyperammonemia type 1. Just remember, there are 2, right? Type 1 and type 2. Just remember this is happening in the first step and with the first enzyme. So hence it's type 1. Okay. Now, what was the next step? Next step was the ornithine was combining with the carbamoyl phosphate to give us citrulline, right? Let's go back over here so that you can relate. Carbamoyl phosphate with ornithine was giving us citrulline. So what did I tell you? The ornithine is not in the mitochondria. Okay, there was a defect in this one. So we'll come back to this. Sorry for confusing you. Let's just focus on this step right now. Ornithine to citrulline. What was the enzyme over here? Ornithine carbamoyl transferase. Okay, so in this one, if this one is deficient, it would cause hyperammonemia type 2. Please take note of this. It's very important. I think they can ask you this because this is the only enzyme which causes an X-linked disorder. Rest all the disorders are recessive. Okay, but this one causes, and this one is majorly present in males, and it is the only one that is, it causes 40% of the disorders in urea cycle. So most commonly seen in males, it causes hyperammonemia type 2. Okay, moving on from citrulline. Again guys, citrulline is combining with what? Aspartate, right? This one. I want you to focus that aspartate is also not present in the cytosol. Okay, aspartate is actually present in the mitochondria, and there's a channel in the membrane that helps to bring it in the cytosol. So that one is called citrin. Okay, so in this one, citrulline is combined with aspartate. Okay, what was the enzyme over here? This one. Arganosuccinate synthetase. Okay, this one. So if it is not there, it would cause citrullinemia type 1. Everyone is following? Are there any confusions? Okay. So next, now we have, okay. So now we have the arganosuccinate, if this enzyme is fine. So arganosuccinate had to undergo lysis, right? Arginine and fumarate. So if this one is not there, this one is really important, guys. The arganosuccinate lyase. It causes arganosuccinic aciduria. Please take note of the following pathologies that are there with the enzymes. I have summarized it in the next slide. This will really help you. Okay. So if this enzyme is fine, arginine is there. But now we want arginine to go back to ornithine and release the urea. But what happened? We don't have the arginase enzyme. So there will be no ornithine, no urea, and it will be causing arginemia because this amino acid will start to build up in the blood. Okay. Now this one, I have even written important. Ornithine, I told you, it's present in the cytoplasm, but it had to go inside the mitochondria to have this reaction. But what happens when it's not going inside the mitochondria? It's a transporter defect. It causes the HHH syndrome. I'll be asking you what happens in this. So please take note. It's due to the ornithine permease as written in Harper's. Don't follow this one. It's due to ornithine permease. Okay, it's a channel defect. It's not an enzyme defect. Please take note. Okay. So these are the questions for you over here. We are almost to the end. Just one more slide left. So metabolic disorders of the urea cycle we discussed. So hyperammonemia type 1, it's type 1. It's the defect in the first enzyme, and the first enzyme is the CPS1. Okay, and it's the most severe one. Can anyone tell me which of the following in 1, 2, 3, 4, and 5, which one would cause the most severe hyperammonemia problem? There are two.
Type 1.
The first step and the second step. Both the steps that are happening in the mitochondria, if any defect is in this one or this one, they would be more severe than any one of this. And the least severe would always be the last step. Because last step was already, arginase is a water soluble molecule, okay? So it would anyway not harm you. I'll tell you this in the other slide, just a second. We'll come to that. I have asked it over here. Okay, so next question. HHH syndrome, what is the deficiency and what is the gene mutation? It is again from your book.
ORNT1 gene.
Yes, ORNT1 is the gene and
For the permease enzyme.
ornithine permease. Yes, please take down note for this one. So, moving on to the main part, I want to explain that why there is hyperornithemia. That means increased ornithine in the blood, hyperammonemia, and homocitrullinuria. Citrullinuria. So sorry. So I told you this thing over here, right, about lysine, this one. So what is happening why are these three things happening? The first one is, so due to the defective transporter, what will happen to ornithine? Will it increase in levels or decrease in levels?
It will increase in the cytosol. It will not, it will not come inside the mitochondria.
Okay, but in the blood, it is increasing, right? There's
Yes.
buildup. So it explains the hyperornithemia. Everyone agrees. Next, why does it cause hyperammonemia? I'm opening the image for you so that you can explain if you want.
Since there is no ornithine, there is no formation of carbamyl phosphate.
No, carbamyl phosphate is anyway forming. But I told you, the first two steps are the most toxic ones, right? So if there is no ornithine, there is carbamyl phosphate is there, but it cannot combine with ornithine to give us citrulline. So carbamyl phosphate levels will start rising because it does not have anything to combine with. Hence we get hyperammonemia because carbamyl phosphate is still a toxic product. It has not been stabilized yet. Okay, next, the most interesting one. Why is there homocitrullinuria? Okay, I'll explain this one. So, since there is a buildup of carbamyl phosphate, there is lysine present in the mitochondria, okay? So when carbamyl phosphate, it combines with lysine, since there is no ornithine for it to combine with, it will form a product called homocitrulline. So since there is no ornithine and it's keep on combining with lysine, it will keep on giving us homocitrulline. So hence now the homocitrulline levels will keep on rising, giving us the homocitrullinuria. And homocitrulline can be excreted in the urine, hence it's nuria. Is everyone clear with that? Did you guys know that? Okay. Okay, cool. So next, moving on to the most important one, hyperammonemia type 2. This was the defect in the second enzyme, ornithine carbamylase. Okay, so it's X-linked and it's found only in males. Okay, next one, arginosuccinic aciduria. It is caused by, what is the defected enzyme in this condition?
Arginosuccinic
arginosuccinate lyase, which was giving us fumarate and arginine. Okay, so please remember all the conditions associated with this. I'm very sure they can ask. Again, it's a question, right? Please take down note. Remember this keyword, trichorexis nodosa. Trichorexis nodosa is nothing but, imagine a hair with nodes in it. It literally looks like two brooms stuck together, you know. So very explicit in the word itself. Okay, so next, hyperarginemia. So it is the defect in the arginase enzyme. I told you all others were the recessive conditions apart from the hyperammonemia type 1. So I want to ask, when do you think symptoms would occur for this condition?
After 2 to 4 years of age.
2 to 4 years.
Yes, but in all other conditions, it's generally seen immediately after birth, or few days or few weeks. But in this one, 2 to 4 years, because I told you that arginine was not so toxic, right? It has already been modified. It just had to release urea. So it was not so toxic. So hence the symptoms appear late. Please take down note that it occurs around 2 to 4 years. Okay. So this is our last slide. I want to just sum it all up. So during the presentation, I told you to note down about this. But first, I told you to find out other cycles that take place in both the cytoplasm and mitochondria. Can someone, I already gave a hint over there. What are the other two pathways that take place in both the cytoplasm and mitochondria? Always remember, hug requires two people. Two people, one person is cytoplasm and one is mitochondria. So here U would be the urea cycle. What is H and what is G? Last slide, guys. Come on, then you can also go study. Google it, it'll come there. Just want you to know because it's very important.
Heme synthesis, gluconeogenesis.
Yes, Wilson. So H stands for heme synthesis pathway, U stands for urea cycle, and G stands for gluconeogenesis. Please take note of this one. So next, during the urea cycle, I kept asking you to take down note of the ATP being consumed. So can someone give me the energetics of the urea cycle? What is direct ATP used and what are the equivalent ATPs being used? I'll give you MCQ. Option 1 is 1, then option 2, 2, option 3 is 3, and option 4 is 4. Tell me for direct first.
3.
Okay, 3. So we saw over here 2 ATPs, 1 ATP. So direct ATPs used are 3. Correct. Next, what are the equivalent ATPs used? Anybody? I'll open this diagram for you so you can correlate. Okay. Go. Tell me the equivalent ATPs used in the urea cycle. And whoever is going to answer, please explain also why are you answering that. Anyone? No one? Okay, I'll answer. Or you want it as an assignment, you're more than welcome. Take this as an
Is it 4?
assignment. Who's answering this one? Raise your hand. I want to ask. Please explain why you're saying 4. Okay, take this as an assignment. You can text me on my TG once you know the answer. It's 4, but if you're able to find out why, okay, yes, iPad, you can tell me why 4.
As you can see in the pathway, the two are consumed in the mitochondria itself,
Okay,
and one is converted to AMP.
exactly. So
uh,
hence it is 4, because here it was only using one high energy phosphate. So 2 molecules gave 2 high energy phosphate, whereas in ATP over here was converting into adenosine monophosphate. So 2 inorganic phosphates were released, high energy ones. So hence the equivalent ATP used is actually 4. Okay, please take note of this and always read the question carefully and see what it says. Does it say direct or equivalent? Okay, next, here's the image of a pediatric patient who came in. Can you tell me what's the diagnosis and name the enzyme deficient? Come on, guys, last question. I'm honestly, this is the last slide and the last question.
Trichorexis
this-
nodosa.
Yes.
Trichohexis
No,
nodosa.
trichorexis nodosa is a manifestation. What is the main
It-
problem?
Arginosuccinic aciduria.
Yes, exactly. So good. Trichorexis nodosa, or you can see tufts of hair. That is a symptom. It is not the diagnosis. Guys, differentiate in those two. So diagnosis is, as Anisha told, it is this one. Arginosuccinic aciduria. And what's the enzyme deficient?
Arginosuccinate.
Lies. Yay. Great job. Okay. So as I promised, that was my last slide. So is everyone clear with the topic? You can open up your cameras, guys. It's okay. Okay, did you get to learn something new? Actually, guys, I had a feedback form. If you can fill it up for me, it will be really helpful so that, I can modify further lectures if you. Okay, if you have like 2 minutes to spare, you can please fill that up. And anyone has any doubts, you can chat me on my TG. I'll give you my number. Okay, there's my TG number. Okay. So Kshitij, you have anything to say?
Thank you. That's all I can say. Thank you so much for revising it. Because like most of us are confused for biochemistry, especially. So
Okay.
like this would have motivated people to like keep studying today night and till tomorrow morning going into the exam. So
Yeah.
that's cool. Thank you so much.
Guys, really a very useful tip. Please make a strong base of your first year subjects. Okay, I personally face this problem, hence I'm telling you, have a strong base with anat, physio, and biochem. I cannot stress enough. If you really do these 3 subjects now, you won't have to put in a lot of effort in second or in third year or in your final year. You can solve everything with these 3 subjects, everything. Okay. And I hope everyone can you show me your sheets? Did you note down the questions I asked you? And let me know if some of them come in your exam tomorrow. So it'll help me. Anyone made the sheet? No one? No one wrote down the questions. Okay, Kshitij did. How about you, Pawan? And Wilson?
I just took the notes of both.
Okay. Okay, guys. So that was it. If you want, I'll share a copy of my presentation with you. I'll give it to Kshitij. So yeah, all the best for your exam tomorrow. Let me know how it went. And that's it. Go study and sleep well. Thank you so much.
Thank you.
Thank you.